Hi everyone, I'm Joelle Eaves from the Woldring Lab at MSU, and today I'll be discussing a paper titled Prediction of Drug Target Affinity Using Attention Neural Network. This paper came out about a year ago, and its intent was to predict the binding affinity between a small molecule drug and some target protein. So this model relies on both the protein sequence and the ligand sequence. The protein sequence is encoded using one-hot encoding into some dimension that is constant for all proteins in their data set. So the proteins in their data set are of varying lengths, and how the authors get around this is by choosing some common size. In this case, they chose 1,000, and any protein that was smaller than 1,000 residues was padded with zeros. So they had a lot of empty features in those smaller protein representations. Any proteins that were larger than 1,000 residues were simply truncated so that the representation only encompassed a portion of the protein. Once they had that target feature matrix representation, they passed it through a bidirectional gated recurrent unit followed by a soft attention layer, and then they pulled those features into some common dimension vector y. As far as the ligand, it was represented in this two-dimensional graph using RD-kit physicochemical properties that were determined from the smiles. That graph was passed through what they call graph sage, and then it was pooled and projected into, again, a common dimension in this vector YD. Both the protein or the target representation and the drug representation were passed to an attention neural net, and this model was used to predict the binding affinities. So they tested their model on two different data sets. The Davis data set is smaller, it contains about 30,000 points, and the Kiba data set is much larger with about 120,000 points. You can see here that the Kiba data set has more unique drug compounds, and the Davis dataset has more unique protein compounds. They show their model's performance, GRA DTA in blue, compared to 5 other fairly state-of-the-art models. We see that it outperforms the other models on the Kiba dataset for sure, though it's less competitive on the smaller Davis dataset. They also conducted some ablation studies. So here you can see their GRA DTA is their entire model, and then you can see with no protein, with no attention neural net, with no protein attention or attention neural net, and then this is the predecessor graph DTA. It's just an older model, and so you can see that their complete model clearly outperforms any sort of ablated model. I highly recommend checking out this paper. Thank you.